Hello and welcome to the Waketree Online Academy. My name is Chris and I'll be your tutor for today. We'll be looking at the Cambridge paper 2.1 for the AS Biology paper, winter 2019, course code 9700. Question 1. Figure 1.1 shows the structure of an amino acid called glycine. Name the parts of the amino acid molecule labeled A and B in figure 1.1. Okay, so this is a simple amino acid structure question. You should know this. A is the amine group. Okay, the mark scheme does allow you to say amino group, but amine group is the correct name for it. Okay, so don't just put amine group because it's a group of things here. And B refers to a carboxyl group, and you can tell this from your early chemistry knowledge as well. So this is called a carboxyl group. Okay, that's an easy 2 marks. You don't want to lose this out on an exam question. Question 2i. Amino acids are monomers used to build proteins. Complete figure 1.2 by drawing a diagram to show the formation of a peptide bond between the 2 molecules. Okay, so in this you can actually use the diagram here. This is a condensation reaction. So what happens here is that this area here will come together and expel water as a product. Okay, so this is the first part. Identifying water as a product is 1 mark. Correctly identifying where the actual bond takes place, and then the final bit to draw it is really the end bonding that takes place here. Okay, so if you were to draw the full amino acid structure like this, that is fine. Okay. Question part 2i. Amino acids are monomers used to build proteins. Okay. Complete figure 1.2 by drawing a diagram to show the formation of a peptide bond between the 2 molecules of glycine. Okay. So if you remember, you should understand that the bond will take place between the hydroxyl group and the H of the amine group. Okay. And that water is a product in this reaction. Okay, so water is expelled from this reaction as plus water as an additional product. Okay, so what you do then is just draw the rest of the diagram to illustrate the full marks. Okay, so you have here, let's just finish drawing our molecule. Okay, and you'll have a bond between the carbon and the nitrogen, and then you draw the rest of the molecule. Okay, and also, and you can put plus H2O here as well, just to highlight the fact that it is a product, and just correctly identify that this area here, okay, is where your peptide bond is taking place. Okay, this is your peptide bond. Okay, excellent. Question B. Plasma cells synthesize and secrete antibodies. Okay. Figure 1.3 is a transmission electromicrograph showing a plasma cell. Okay. Use the label line and the label T on figure 1.3 to identify where the genes for coding the polypeptide chains are located. Okay. Well, as you know, these would take place in the nucleus. Okay, so anywhere in the dark shaded area with the label T. Okay, remember it's a straight line, no arrows, using a ruler, and you have to use the label T. An easy 1 mark. Question part 2i. Calculate the actual diameter of the plasma cell shown in the line P to Q. Okay, write down the formula used to make your calculation. Show your working and give your answer to the nearest micrometer. Okay, now it clearly gives you an instruction. Okay, so it's worth 2 marks, and it says write the formula down. So this is indicative of that this is part of the question too, and worth 1 mark. So if we remember from our earlier formulas in GCSE and A level, you remember this near kind of triangle. You just rearrange it so A equals I divided by M. Okay, so you can put A equals I divided by M. Okay, give your answer to the nearest micrometer. Okay, so what you'd do is you'd use a ruler, measure from P to Q. this length is roughly around about 9 centimeters equals I. Okay, but before you do this equation, what you should do is really convert it into micrometers. So first convert it into millimeters by timesing 10, and then times it by 1,000 to give your answer to 90,000 micrometers. Okay, so then what I would do is I'd do 90,000 divided by 6,000, okay, and do the maths. Okay, and then it's effectively 90 divided by 6, which gives us the answer of 15 micrometers. Okay, be careful on the unit conversions here. This is where most students lose the marks, but this is an easy 2 marks in an exam you should be able to do. Part 3I, the plasma cell in figure 1.3 is very metabolically active. Suggests why there are very few mitochondria visible in the electron micrograph shown in figure 1.3. Okay, well, mitochondria are moving around quite a lot inside the cell. Maybe they're not in a position to be seen. So instead of being horizontal for them to be picked up by the electron microscope, they might be vertically. So that way you won't be able to physically see them under the microscope. Okay, so, you know, this is only a very small section of the cell. Okay, maybe in other parts of cell. Yeah. You could also mention that they're hidden behind other organelles, or they could be behind the nucleus or something like that. Any valid suggestion to why they're not visible. But one of the answers you shouldn't say is that they can't be seen by this microscope. Well, yes, they can. Although they're very small, the electron microscope can pick up mitochondria at various magnifications. Okay, so you have to think of another reason other than the fact that they're just the limitations of the equipment, because in this case, it's not one of those marks. Okay. Question C. Sieve tube elements in plants have very few organelles such as mitochondria. Explain how having very few organelles is an adaptation of a sieve tube element to its function. Okay, well, the phloem's main function is transportation by translocation. So having organelles gets in the way. Okay, so having less organelles, okay, reduces resistance. Okay, reduces resistance of flow. Okay, now not only that, but the phloem itself doesn't actually do the active loading or unloading as such. Remember that there's a companion cell attached to the phloem that does all the main work, and they would have all the organelles in there as well. So they have companion cells, okay, companion cells that contain most organelles to support its function, e.g. the companion cells would do active loading, okay, rather than the actual phloem cells themselves. Okay, so these 2 answers are perfectly acceptable here. There are other points, but this should give you the most marks easily. Thank you for watching our video today. Please register for a free demo on our website, and I look forward to seeing you in our next video.